Hello, everyone. It's my pleasure to be here talking about CRISPR gene editing technology. I'm Dr. Youwei Zhang, Asian scientist from Ubigene Biosciences. Today, I'm going to share with you the genome-wide CRISPR library screening. Here's what we are talking about. We have four parts. First, I will give you the basis on CRISPR screening. Next, I will break down how the screening process works and point out the key steps that really matter. Then, we will explore the different ways CRISPR screening can be used. And finally, I will introduce Ubigene CRISPR library screening and how it might accelerate your research. So first question, what is CRISPR screening? CRISPR screening is a large-scale function gene screening. It is a powerful technique that uses a collection of CRISPR guide RNAs to systematically edit and investigate the function of thousands of genes in parallel, enabling the identification of genes involved in specific biological process or phenotypes. Another question, why should I do CRISPR library screening? Okay, let me break it down for you. So usually we can find target using multi-omics method, right? But why do we use CRISPR screening? So compared to traditional multi-omics methods, CRISPR screening offers several key advantages. First, direct function assessment. CRISPR screening directly edits genes, providing clear causal links between gene function and phenotypes. Traditional omics often requires additional function validation. Second, high-throughput functional validation. CRISPR screening identifies genes involved in biological process or disease, even those with unknown functions, efficiently supporting large-scale functional studies. So third, target operability. CRISPR screening can really nail down gene targets that you can treat right away for therapy, like fixing gene in monogenic disease, but traditional omics usually gives you targets that need a lot of more work and a complex process to develop drugs. So fourth, the low noise and high efficiency. CRISPR screening, especially CRISPR-I, has lower off-target effects and higher editing efficiency compared to RNAi, leading to more reliable results. So fifth, the flexibility and customization. CRISPR library can be easily customized for specific gene sets or biological context, and the screening process can be adapted to various experimental challenges, such as the drug treatment or infections. Finally, for the cost efficient, so CRISPR screening can be more cost effective, especially in large-scale experiments, compared to the complex and resource-intensive nature of traditional omics analysis. Overall, CRISPR screening directly links gene function to phenotypes, efficiently identifies actionable targets, and provides a flexible and cost-effective solution for large-scale function genomic studies. That's why we choose CRISPR screening. So, we are familiar with the CRISPR knockout library, right? Well, by tricking the Cas9 protein cutting ability, we can also create libraries in the lab that an easy boost or block gene expression. We call this CRISPR-A or CRISPR-I libraries. For the CRISPR knockout library, the guide RNA in those libraries mainly target the 5' exon of coding genes. The double-strand break induced by Cas9 are repaired by the NHEJ mechanism, leading to frame shift mutations and gene knockout. So for the CRISPR-A library, this library targets transcription regulatory regions of coding and no-coding genes. By linking Cas9 with transcription activator, such as the VP64 and co-expression with MS2, P65, HSF1, it will efficiently upregulate genome-wide expression for high-throughput gain-of-function screening. When we study the non-coding and essential gene, we can use the CRISPR-I library called transfectin inactivated Cas9 fused with the transcription repressor such as KRAB and guide RNA targeting upstream regulatory region or achieve gene knockdown. So Which library you pick just depends on what you are trying to study. Of course, you can use both CRISPR knockout and CRISPR-A libraries together to hunt down target gene. And how does CRISPR screening work? Okay, let me walk you through the process for CRISPR library screening. Let's say we are trying to find a drug target, like looking for genes that make cells sensitive or resistant to a drug. To make it simple, imagine we have built a small 4D library. Say we knock out 6 genes. We make this guide RNA, build the plasmid library, package it into lentivirus, and then get that virus with Cas9 and the guide RNA into the target cells. After drug selection, we got a pool of cells that stably express the knockout guide RNA. Now, let's say after a couple of weeks of the growing these cells, we notice that with the cell gene knockout just disappear. That tells us C is a must-have gene for the cell survival. So we'll find an essential gene just by seeing which cell don't make it. We check the cells after 2 weeks and split them into groups, like a control group and a group with drug. After treating with the drug and sequencing the cell, we see that cells with the B gene knockout are gone compared to the control. That means B is a resistant gene. The drug can't touch cells without it. So on the flip side, we might see that cells with A gene knockout are way more common than in the control. That means A is a sensitive gene. The drug really hits those cells hard. A could be target we are looking for. So we have got 2 ways to screen. Negative screen, where we look for the cells that disappear, and the positive screen, where we look for cells that pile up. Either way, we can find the potential drug target. Once we have got our targets, we can dig deeper to figure out the pathway the drug is hitting. So how can we get started with our CRISPR library screening? Firstly, we need to decide on the library we are going to use. There are several factors to consider. First, where is the library come from? Is it an off-the-shelf option from a company, or do we need to have its customer serialized? And second, what's the goal of the experiment? Are we looking at a knockout, an activation, or an inhibition library? And third, what's the coverage of the library? Is it a whole genome library or a sub-library targeting specific gene or pathways? And importantly, is the library targeting human cells, mouse cells, or something else? And it is also crucial to figure out how many guide RNA we are going to design for each gene. Typically, we recommend around 3 to 10 guide RNA per gene for effective targeting. Once we have got our library sorted out, we need to think about the cells we are going to use. CRISPR library screening usually involves a large number of cells. So for the cancer cells, immortalized cells, stem cells, and iPS cells are all good candidates for library screening experiments. For some groups, they may be interested in the primary cells. So can we do the CRISPR screening using the primary cells? Answer is yes. So for the primary cells, which can be a bit trickier, we need to make sure we can get enough cells, and then we have done some preliminary experiment to optimize the screening conditions. But with the right preparation, we can definitely use the primary cells for CRISPR screening and find the target gene. So after constructing the cell pool, the next step is to decide on screening methods to identify target gene. Here are 3 common screening methods: viability screen, report screen, and behavior screen. Generally, the most common method is to screen based on cell viability. For example, after passing gene or drug treatment, cell may experience alteration in their viability or mortality. By collecting the surviving cell, we can analyze the target gene. So if the screening phenotype is not related to cell viability, we can also use fluorescence-activated cell sorting. For instance, When studying the regulator of a signal pathway, we can construct cells with fluorescence protein tagged marker protein. After passing genes, cells can be sorted based on the intensity of the fluorescence signal. By comparing the difference between cells with stronger and weaker fluorescence signals, we can identify target gene. This method is also applicable for studying membrane proteins, where antibodies can be used for sorting to find target gene. So when studying the gene related to tumor cell migration, we can use the transfer culture method to identify target gene. Now, we are choosing our library and identified our cell line and set on our screening method. Now we are ready to start our CRISPR screening. The process included following steps. First, we design the guide RNA and then synthesize them. After that, we construct the library. If a commercial company offers the targeted library, we can also just buy the relevant plasmid for our experiment. Once the plasmid is ready, we package it into lentivirus and infect the target cell with it. After antibody selection, we get a cell pool that stably expresses the library guide RNA and Cas9. This cell pool is then subject to specific screening conditions to isolate the cell of interest. Next, we extract genomic DNA from these cells, amplify this by PCR, and construct a sequencing library for sequencing. So finally, we analyze the sequencing results through bioinformatics to identify candidate target genes. There are several key steps in the CRISPR screening process, such as constructing the CRISPR library plasmid, infecting the cell with virus, and analyzing the sequencing result through bioinformatics are especially critical. So when performing CRISPR library in the lab, we may encounter some challenges, such as difficulty to construct the CRISPR library plasmid, the coverage of the library is not high enough, or the library uniformity is not good, or it's difficult to identify target genes, or maybe sometimes it's difficult to analyze the NGS sequencing data. So next, I will share the experience our team have accumulated in CRISPR library screening. So let's start by talking about the first key step in CRISPR screening, constructing the CRISPR plasmid library. There are two crucial parameters here. The first is the coverage of the plasmid library. This means the proportion of the actual guide RNA in the constructed library compared to the theoretical number we designed. For example, if we design 10,000 guide RNA and the sequencing shows that our library contains 9,000 of them, that's coverage rate of 90%. Ideally, the higher the coverage, the better. So typically, we require at least 90% coverage to proceed with the next step. But our company set a higher standard. We won't move forward unless the coverage is above 99.9%. So the second parameter is the uniformity of the plasmid library. Think of it this way. If we have a library with different guide RNA, we want each guide RNA to be present in roughly the same amount. The smaller the difference in abundance, the better the uniformity and higher the quality of the library. Generally, a library is considered usable if the uniformity parameter is less than 15. Our company standard is even stricter. We aim for parameter to be less than 10, and our company constructs a high-quality CRISPR plasmid library with coverage above 99.9% and uniformity less than 10. Our library that meet this standard are good enough for the downstream experiment. Okay, let's dive a little deeper into key points of virus transduction, especially focusing on the two crucial aspects, MOI and antibiotic selection. First, let's talk about MOI. MOI stands for the multiplicity of the infection. It's essentially a ratio that tells us how many virus particles we are adding relative to the number of cells. This ratio is crucial because it determines how effectively our cells will be transduced. To put it simplified, if you have two cells and only one virus particle, your MOI is 0.5. But if you have one cell and three virus particles, your MOI is 3. It's a straightforward ratio, but it's incredibly important for ensuring consistency across experiments. You want to make sure you are using the same amount of virus each time to keep your experiment controlled. So next, let's discuss antibody selection. For instance, we often use pyrimidine to select stable expressed guide-RNA cells. Imagine we transduced our cells population, but not all cells get infected. Some remain untransduced. They are the risk-free cells. By applying pyrimidine, we can selectively eliminate those untransduced cells. This leaves us with a pure population of transduced cells. This step is better because it ensures that every cell in your culture has undergone the designed genetic modification. And here is another useful feature: fluorescence marker. Even though we have antibiotic selection to ensure our cells are transduced, it's always helpful to have a visual confirmation. Many plasmids include a fluorescence marker alongside the gene of interest. If the gene is being expressed, the fluorescence marker will also be expressed. This allows you to visually inspect your cells under a microscope and see exactly what percentage of them are successfully transduced. In summary, understanding MOI and using antibiotic selection and fluorescence marker are all essential for successful and consistent measure transduction experiment. They help you optimize your transduction efficiency and ensure that all your cells are properly modified. So how can we determine the MOI for our experiment and why it's important? As we know, CRISPR screening requires that each cell be infected by one viral particle, allowing for the editing of just one gene per cell. This is crucial because it enables us to accurately correlate the genotype with phenotype during the subsequent functional screening. Therefore, when constructing the library cells, the virus infection efficiency must not be too high. Research has shown that the higher the infection efficiency, the more virus particles enter each cell. When the infection efficiency is only 5%, the majority of cells are infected by just one virus particle. However, as the infection efficiency increases, by the time it reaches 95%, over 80% of the cells are infected by more than 2 viral particles. So controlling virus infection efficiency to achieve effective CRISPR screening is a critical step. So when infection efficiency is below 5%, the number of surviving cells after drug selection is too low to meet the requirements for laboratory screening. To balance experimental feasibility with the demands of CRISPR laboratory screening, it is generally recommended to control the infection efficiency between 20% to 40%. Based on our experience, an infection efficiency of around 30% is optimal. This ensures that most cells are infected by only one virus particle, while also providing a sufficient cells for the subsequent screening experiment. So another key point in CRISPR screening is determining the coverage of the library plasmid, or what we call the cell coverage. The cell coverage can be understood as the average number of cells per guide RNA in a CRISPR library screening. Research has shown that when cell coverage is low, for example, at 50, the guide RNA in the library cells are lost significantly after a few passages, and the cell coverage dropped sharply. However, when cell coverage is at 500, even after 8 passages, the cell coverage remains high. But as the coverage increases, the number of cells you have to work with also grows. Based on our experience, a cell coverage of 300 is sufficient for the whole general library screening. The choice of cell coverage should also think about the size of library you used. So when using a smaller CRISPR library, you can choose a higher cell coverage. So, okay, now let's talk about the NG sequencing result analysis. So here are some simple data. There are over gene And the guides we used, we got the PGP, MPC1, and so on. As we mentioned earlier, you need at least 3 guides per gene. Here we got 4 guides for each gene. Now, if we compared our replicates, we can see something interesting. For example, PGP is generally depleted across all 4 groups. But check this out. One of these guides didn't perform very well. It's kind of low across the board. So that's exactly why we need replicate. The other 3 guides show really effect. Even so, this one didn't work so well, so we can sort right off of this guide because it's not really working. We also see depletion for a couple of other genes. And then there are these 3 genes that are enriched over time. So having replicates both in our condition and in our guides is really important. It helps us see if there's a true effect. Now, let's review one more time. What do depletion and enrichment tell us about the gene? Depleted genes, they are important for cell growth. When we knock them out, the cell die, so the guides get depleted. So on the other hand, enriched genes are usually tumor suppressor. If this gene were working normally, they'd keep cell growth in check. But since we knocked them out, the cells are expanding exclusive, and the guide get enriched. So after we do the bioinformatic analysis on our sequencing data, we got this list of genes, all ranked by their score from highest to lowest. For the genes that really stand out, either because they are enriched or depleted, we dig deeper. We use things like KEGG and GO analysis to figure out which signaling pathway and metabolic routes these genes are involved in. This helps us narrow down the genes we want to focus on for the further validation. Our bioinformatics team can whip up some really nice visual based on what you need, like those fancy volcano plots or scatter plots you see in top-tier journal. Yeah, we can do that. Take this volcano plot, for example. The genes marked in red are the enriched ones, and those in blue are the depleted ones. This part showed some of the genes with the highest scores, and we can customize them just the way you want. So, once we got the gene list, the next step is to verify the gene function on the target. So we can do this by using the CRISPR-Cas9 system to knock out the target gene or by using the RNAi to knock down its expression. This helps us to see the phenotypic trait of the gene again. And our company offers the full service knockout or knockdown. We got you covered from start to finish. Alright, so we have been seeing that the key to CRISPR screening working well is all about getting the library plasmid right and have a good cell pool. But how did our team actually do it? What's our secret sauce? Our R&D team has their own special know-how and techniques. We have developed super efficient competent cells for transformation. When we construct our plasmid library, we use the electroporation combined with those high-efficiency competent cells. This way, we get really high-quality library plasmid with over 99% coverage and uniformity on the 10. And we will fine-tune our process and have this exclusive method for making cell pools. We can scale up and standardize the production of these cell pools. This means we get super consistent result with minimal bench-to-bench difference and great reproducibility. And the coverage of our cell pool library is up to 99%. Okay, so now let's talk about the application of CRISPR library screening. CRISPR library screening is a powerful tool for understanding genes. So what can CRISPR screening do? Here's what it can do. Firstly, it can give you the big picture of gene study. You can trick lots of genes all at once and figure out which one are the real deal in disease or cell behavior. And helping to find drug targets It helps us pinpoint genes that could be perfect targets for new medicines. So moreover, understanding the pathways. After we edit the gene, we can see how cell change and figure out where these genes fit into the bigger picture. What's more, it can also help us to making the disease model. We can trick cell to act like these have disease and study them up close. It's really revolutionary with the way we explore the gene function. And I wanted to talk a little bit more about the application of CRISPR library screening. So here are the examples for the cancer research. This powerful technology is revolutionizing the way we approach cancer research and treatment. CRISPR screening enables us to identify novel drug targets in cancer and develop treatment strategy that specifically target tumor suppressor mechanism or loss function pathways. It facilitates the exploration of biomarkers that predict drug response and allow us to characterize the off-target effects of existing drug more accurately. So we can also investigate the regulator of the drug resistance and develop innovative combination therapy to effectively overcome resistance. Additionally, it supports the study of immune cell function regulators and assess their potential as target for the immunotherapy. So in summary, CRISPR screening opening up new avenues for drug discovery, improving our understanding of the cancer biology, and bringing us closer to more effective and personalized treatments. So the application of CRISPR screening is becoming increasingly popular. We are seeing more and more high-impact articles using CRISPR screening methods to identify target genes. For example, positive screening is used to search for drug targets. Negative screening is employed to find regulatory factors. There are also studies that use the flow cytometry sorting to identify anti-tumor genes, or apply the visual infection pressure to screen for the host gene-related antiviral response. To sum up, CRISPR screening is such a powerful tool that its use are just gonna keep growing and spreading to more and more areas. Okay, so this is our company's ready-to-use CRISPR library product, including the whole genome knockout activation or inhibition library, more than certified CRISPR library, plasmid, and virus, as well as over 400 CRISPR cell library. So making genome editing easier is our goal. Our company has provided service and product to customers from more than 40 countries. So our company's business include CRISPR libraries, gene editing cell, and gene-modified micrograms. So far, our collaborators have published over 200 papers with our help. And we are looking forward to empowering your next breakthrough. Thank you for your listening. That's all.